I'm happy to introduce Jim DiCarlo. You know him already from my talk on Friday, because I mentioned that he's the director of the Quest, who is essentially carrying forwards the vision of the old CBMM, and is also the co-director of CBMM with me. So I'm proud to say that I think I was one of the people who brought Jim to MIT, and there is a long tradition in neuroscience that the visual system has been one of the areas essentially marking and demonstrating progress in our understanding of the brain. And this is at least since the time of Hubel and Wiesel in the 60s. And I think Jim is, the continuation of this tradition, and our best hope to have new breakthroughs on how the visual system works. We disagree sometime about the details of the approach, and it would be interesting to discuss about those things in the next few days. But he has been doing great work, and will continue to be great work in the future. I hope in some revolutionary breakthroughs. One of the most interesting things in science, in the history of science, is what was called scientific revolutions, like what happened when quantum mechanics changed the picture of physics, and the same for relativity and so on. It would be great if this would happen in neuroscience, and if neuroscience, as I mentioned in an implicit way on Friday, would at some point in the near future drive also artificial intelligence rather than the opposite. So with this, Jim.
So I'm going to start by I'm going to say a little bit, and this relates a little bit to what Tommy just referred to, and I think it'd be fun to talk, as you see throughout the talk, of like where there's disagreements on how we should be proceeding, and I've inserted some of that in the talk, and maybe that will come out in discussion too. But I titled my talk, "The Quest to Create Engineer Quality Models of the Mechanisms of Human Visual Recognition," which I view the part of our job here is to build an understanding which will look like something engineers could transmit to other engineers. And I think that's in the spirit of CBMM, really vision. And I'm going to tell you about that within the scope of visual object recognition. So there's a bit of kind of what's going on in that space, a bit of history. If you don't know that, if you do, forgive me if you've seen this story, but I think most of you are maybe new to the field a bit, so I'm going to introduce you to that. You'll notice a bit of scientific philosophy weaved into all of this as well, and I'll try to call that out as well. And that relates to this, sometimes I call this reverse engineering, not all of visual intelligence, but at least the core component of visual intelligence. And this term reverse engineering is something that Tommy and I debate, is this really what science is about? But this is what I mean by reverse engineering, is discovering models or building models of this type, which I view as scientific hypotheses of how the system might work. Okay, so let me start by motivating you a bit. Like, why are you here in the course? Why are you even thinking about the questions of Center for Brains, Minds, and Machines, or intelligence broadly? Okay, so I'm going to motivate this by showing you a video from Hans Lucas Teuber, who was the chair of the Department of Psychology, which is now called Brain and Cognitive Sciences, which was a department that Tommy and I are both faculty in at MIT, and that I chaired for 9 years. So I had his job after him. But I'm going to show you this just for context here, because it was partly what motivated me, and I think Tommy and others in the field. And Teuber saw sort of what the hard questions were often not the ones that you would have imagined coming into the field of psychology at the time. And so I think it's a good motivation of that, and at least tell you what was motivating him, and I think our department more broadly, and I think CBMM as well. So here, let me take a listen. So this is 1974, Hans Lucas Teuber.
In a way, we could say that the first step in any new field is to know what to be astonished at, what to wonder at. A very, very gifted theoretical physicist by name of Murray Gell-Mann, who was here on a visit from that strange place on the West Coast, Caltech, our competitors. We tried very hard to persuade him not to go back there, but we failed. Gelman said to me, without any provocation on my part, that he thought ours was a weak-kneed department, rather than being unorthodox, as I claim, because it had in it neuroanatomy and neurophysiology and linguistics and other things that are usually left out. It was, not bold enough, because we were not studying those issues in psychology that were really important. I said, which ones are important? And he said, and he absolutely floored me by saying that, because I think he's bright. I know he's bright. He said, well, you shouldn't study perception. You should study extrasensory perception. You should not study language, but thought transference and hypnosis. And there was one third thing, which I have forgotten or repressed, as some people say. It might come back under hypnosis, maybe. But in any case, I was astounded and shocked because he said all this in front of our then provost, fortunately a very good friend of mine. I tried to convey to him the idea that the thing to wonder about is perception, that if extrasensory perception did exist, I would feel somewhat let down. I'm not against its existing, but I would feel deflated because it would be much easier to understand. Perception is a great riddle. How do I see a line as a line? And a face as a face? And as that face amongst 1,000 faces where emotion and memory enter? That's a much greater riddle.
Okay, so he's really motivating it. At the time, psychology was, as you heard, some of those things there about repression and so forth, but just even core issues of how we see, or what he's calling perception, is a fascinating question, still is a fascinating question. So that was what astonished him in the department. You also heard him saying, we're bringing together neuroanatomy, neurophysiology, sort of natural science. He didn't mention it, but I think in the modern area, along with computational techniques to interface those. But really, there's a question there, which is how does this thing work that we just take for granted that we see? Language, perception. So one thing, don't forget about how fundamental and hard those problems are that we all just take for granted every day, including just the ability to recognize objects in the world, which is the thing I'm going to tell you about today. Okay, so visual intelligence is astonishing. You look and say, okay, there's cars, there's trees, there's people. What's in the scene? Where are the cars? Where are the people? These are questions you can answer instantly, and even a child can answer. So they feel like they should be computationally trivial, although they're not. The other thing is more complicated visual intelligence questions like what will happen next? Where is it safe to walk? These are sort of forward prediction kind of tasks of visual intelligence, and so on and so forth. Lots that you can do from just this image, right? And all of that is still, much of it is still mysterious, but still astonishing. And we're starting to make some inroads into how we can have engineered models of how our brain accomplishes at least some of those things a bit on the left. And that's what I'm going to tell you about today. So just setting context, you should be motivated by these questions, but knowing we don't have all the answers, hopefully you will help us get the answers, but telling you a way forward to show you how at least we've made progress on some of these first things on the left here. This is another way of saying what I just said. If you think of all of human intelligence, it's a vast open space for science to still understand. How does that work? I'm going to tell you today about just the first couple of 100 milliseconds of questions around estimating what objects are in the world. I call that core recognition. There are other things that happen very quickly around other sensory modalities here that I'm not going to talk about. But broadly, we think this is a foundation for more general scene understanding and intuitive physics and so forth. You'll hear about this especially from Gabriel Kryman, from Josh Tenenbaum, Nancy Kanwisher, and others later in the course. I think this is partly why I'm at the beginning with sort of like how do we just get started on some of the core aspects of vision and show how we can bring engineered models into that game. Okay, but just keep in mind that if you're interested in all these things, that there's sort of a foundation on which all of this is ultimately built upon. I hope we can think about the philosophy of what I'm going to show you coming into all of that in terms of models meeting data, but just keep this in mind from a kind of, I'm not going to tell you about all of visual intelligence. I'm going to tell you about one aspect, a core component of it. Okay, again, don't be afraid to ask questions here. So, okay, that's the human brain. This is a quote I like to give to sort of motivate you also about kind of what the form of the understanding might look like. So this is a quote from Francis Crick, who is the late Francis Crick, co-discovered the structure of DNA. Here's a quote. You, your joys, your sorrows, your memories, your ambitions, your sense of personal identity and free will are in fact no more than the behavior of a vast assembly of nerve cells and their associated molecules. Now, if you think about that question, it's sort of, in one sense, it's sort of inspiring because it's like, wow, this is like, it's possible to understand this. Also a little bit humbling to say, well, I thought we were more than that. You know, I have a soul and all that, but this is saying we're kind of a machine. But this is actually the central dogma of neuroscience, or one of the central dogmas. Yes, we are a machine that can be understood in terms of its parts. Or this sort of no more than, it's sort of somehow we're like dramatically more than. Somehow all those parts work together to make us do much more than those individual parts. And I think that's sort of illustrated. I like to illustrate that here. If you think about an engineered system like this one, that we actually know how it works. Here's another quote. Your remarkable favorite app, all its performance and amazing user interface are in fact no more than the behavior of a vast assembly of transistors. Now, so those of you who think about engineered systems like this, it's like that statement's true, but sort of very insufficient, right? So what's missing here? So I call this the true but not yet useful hypothesis, right? So and this anonymous quote, I read this up, so I'm the anonymous one. So I'm just trying to say, like, kind of get the connection here. These are both true, but also sort of motivating, like, we got to figure out something else here, right? So I think you guys already know this, but let me again give you this is a bit of philosophy again. So what are the kind of answers that we seek? Well, the amazing phenomena of human cognition, whether it's visual intelligence, navigation, memory, all the other kind of aspects of intelligence I was loosely motivating there, they're somehow emergent from a specific set of mechanisms. At least this is how I like to think of it. Some set of a specific neural network plus your body is what's making you do all these intelligent things. So it's not, in that sense, it's not a mystery. It's not there's a dualist perspective and there's a soul and things we'll never understand. There are a set of mechanisms, just like the phone has a set of mechanisms, but they're a complicated set of mechanisms that will be challenging for us to understand, but are understandable in this engineering sense. And so can ultimately be understood in the same way that phenomena of any engineered complex machine are understood. So again, I like to think about the analogy here. How do we understand as a species how an iPhone works? Like, what does the form of that understanding look like? It's not a few simple equations. It's very complicated, transmissions between engineers of how the different levels fit together and how they give rise to the whole system. And in fact, I also like to remind students that no individual human really understands deeply how an iPhone works, even though we built it, right? So it's collective knowledge that we're building up together that has to be managed in a way that's not just words and abstracts on papers, but sort of managed code or managed specs on engineering sheets that are going to have to manage to actually build this kind of understanding. So this, I know Tommy can jump in at any point if there's parts I disagree with, but this is sort of related to when I say reverse engineering. This is kind of what I have in mind. It's like there's an understanding that's going to be a little unsatisfying in that same way. In the same way understanding an iPhone is maybe not completely satisfying. So I think I just said this: reproducible engineered quality specifications of how the component parts interact to produce the emergent phenomena, in this case, the intelligent appearing behavior that I motivated. And I call that spec a system model. So somehow we need complicated models that go from the parts, neurons and their connections, up to the emergent behavior. And I'm just going to refer to that loosely as system model. And I don't mean just today's ANNs. I'm going to show you some ANNs, but I mean some kind of spec that looks like, that can specify this in a way that's transmittable to other humans. Yes, question. When you say my approach, I think what you described is the approach to science in general. And Mar, of course, Mar is set up very nice ways. So Mar and Poggio set up very nice ways to think about systems need to be understood at all those levels. The only part I might disagree with is like there's often not clean breaks between those three levels. And really there's sort of a way of phrasing exactly as you said, like, okay, here's a general idea of what's going on. Okay, how do we dig deeper? How do we dig deeper? How do we dig deeper? What do those next levels look like? And what I'm describing here is trying to dig deeper. Then I could say, the temporal lobe solves object recognition like my iPhone does its app, let's all go home. Now, that's not useful to an engineer. It's also not useful to a clinician trying to help you if you have a temporal lobe disorder, right? So it's sort of that spirit in which we got to be careful with the word understand, because it implies we're finished. And you may understand in your own sense of, oh, I think I understand what iPhone is. But when you then sit down with the engineers from Mars say, help me build a new iPhone. You're like, well, it kind of does this, right? You have to get down to another level to actually do that. So I'm not disagreeing with you, but I'm kind of trying to, I tell my students, try to say what you want to do without using the word understand, which is a cover word for not actually getting to this kind of detail. So I would prefer sort of emulate or build as a kind of goal to sort of demonstrate our understanding. So I'll say more about that later. Explain, predict, and control are sort of ways to phrase science. I'm going to show you a slide from Tori Relay that gets into that. Do you want to push back on that a little bit, or should I keep going? Let's keep going. Okay. But this is exactly a good discussion, just getting into the philosophy of how we approach systems in general. Okay, here's the iPhone example. Those of you who know iPhones, right? Of course, there's transistors, but there's lots of levels that have been defined by engineers that helps us maintain that knowledge. Of course, we know the brain is made of neurons and their synapses, so billions of neurons and trillions of connections that somehow are organized into circuits and systems that live in different brain regions that are somehow executing data and state algorithms, as we'd like to describe it, that give rise to the emergent phenomena of behavior. Now, I don't mean to imply that the brain is an iPhone or that this maps to that or anything like that. It's just to say that there are sort of multiple levels here. And partly, it's not clear that biology is going to leave us nice, clean breaks here, although engineering had to do that, partly to manage it. That's one of our challenges, to try to even figure out what the right levels to put here on the bottom might be. And a lot of the models I'm going to show you today are sort of attempts to go essentially from neurons all the way to behavior with a little bit of this stuff in it here. But again, the main motivation here is to tell you this is sort of a multi-level sort of problem, which again, a system model is meant to bridge this here, link across these scales, at least from neurons in this case to behavior. Now notice I could add levels on here, molecules that maintain neurons, DNA, I can go down to physics. There's other levels that I'm not showing you. So this is sort of choosing a start point and then going to here. That's not to deny the other points of interest, and that's another good topic to talk about. But just to say, what I'm going to try to tell you is that neurons to behavior and trying to build models that cross that bridge. Okay. So this is what I just said. So I'm going to flip through it. So try to account for the ability of the mind in terms of the components of the brain and the language of engineering, which are predictive models. And I sometimes refer to those as sort of possibly intelligence algorithms or really engineering specs. And the science of providing measurements and discoveries of what's in the mind and the brain. And engineers are building things that I view as scientific hypotheses, and there's an interesting meeting ground right now, which is some of the engineered models become our hypothesis, our field, and our job is to test them and also to inform their direction, to build the models in a different direction, maybe not in the direction they're being built. So there's interplay between these fields around system models is how I think of the productive form of this working as a field. Okay, so really what this means is specific in silico, in this case, neural networks, because they're trying to go from neurons to behavior that actually work. That is, and can actually explain the empirical data. And by that, I don't just mean the neural data, but also the behavioral data. If you think about if those networks work at the behavioral level, then in some sense they work scientifically. They're also working in AI kind of applications, at least ones that you want to emulate human abilities on. That's what I mean by work. I don't just mean fit one neuron. I mean explain the whole system. Okay, from at the neural level to the behavioral level. All right, so that's the general philosophy. So today I'm going to try to review this for you, this general approach in the area of visual intelligence, or broadly what Teuber called perception, and maybe give you a motivation of why this matters at all. When I say we, I'm going to show you stuff mostly from my lab. This is kind of our current lab team, and I'll try to give acknowledgement. Some of them are in the audience. Ko is here, and some of his work I'll show you. I'll try to introduce or mention them along the way, but I'm just an ambassador. Anything that was done in the lab is really accredited to these amazing women and men, and I just get to have the fun part of talking about it. Okay. So back to visual intelligence. As I mentioned, we're working on these first parts here on the left. I'm not going to tell you all of visual intelligence, so let's discuss that again at the end. But I'm going to show you when you operationalize a problem, then you can start to make progress on it. So what we operationalized early on, and really this is way back for us before even 2000, as Tommy mentioned, the field in this area is really decades old. But the way we were approaching it when I started my group is to just think about, okay, as a primate, when you're digesting information on the scene here, like the scene I showed you earlier, which you see faded in the background, if you were fixating there, that is, your eyes are there, we know from biology that you have most of your retinal acuity at the center of gaze. So this is meant to illustrate about 10 degrees of visual angle. 10 degrees, for those of you guys who don't know degrees, one finger is like 1 degree. So 2 hands at arm's length is about 10 degrees. Okay, so that's just, think about just that. It's not your whole visual field, but it's sort of, it's where you have very high acuity vision. And our goal is not to solve all the vision in the entire thing. That is a long-term goal, but just to focus on what do you do in that central 10 degrees where you're gathering information right here. In this case, happens to be sort of one kind of major object in the foreground. But as you notice that you don't just dwell there. When you digest a scene, when I flash this up, you didn't realize you were doing this, or maybe you did, you're doing it subconsciously. You're making eye movements around the scene that might look like this. You're fixating, and then you're moving. This is called a saccade. You're fixating, and you're moving. Gabriel Kremen will tell you a lot more about these kind of patterns. You dwell. Anybody know how long you tend to dwell? Or maybe I already have it on the slide. It's about 200 milliseconds or maybe a little bit longer, just your natural. And you don't notice you're doing this, but if you look at a friend while they're looking at a scene, this is what's going on. You see here, you don't see here. Your eyes are moving too fast for you to take in information or the retina to process it. So you get a glimpse, process, get a glimpse, process, get a glimpse, process, right? So that was a framing of which biology is like, that's how primate vision works. And so we use that to set up a problem that we call core recognition, which is the ability to identify or categorize an object, and maybe it's other latent variables like pose and its position and so forth, in just a 200 millisecond glimpse like this one. These are now me simulating your eye movements without you moving. So if you fixate here, I'll just show you glimpses from that scene. Okay, so that's what's coming to your retina, the center of your retina, when you execute a sequence like the one I just showed you. And I noticed there's sort of like each image is on for about 200 milliseconds. I hope you can notice you can identify, and this has gone a couple times, so maybe it's not quite the same, but you can identify usually one or two objects in those glimpses. Okay, that ability to identify one or two of the foreground objects in that glimpse is what we refer to as core recognition, central 10 degrees, 200 milliseconds or so of duration. It's just an operationalization of related to primate vision that we use that we refer to as core recognition. Just a way to get started on the problem. And again, the questions that we wanted to ask is what's here? Like, is a car here or not? In this case, the answer is yes. And in the next frame, the answer was no. Is a person here? Is a sign here? And as I said, I call this core recognition. I've already sort of given you this kind of operationalization of that. And again, we do this partly as a way to get started. So now we can start to test systems in these regimes and compare hypotheses, i.e. models, with natural systems. In this case, I'm going to show you monkeys and humans. You guys with me so far? So sort of narrowed vision to a smaller space. Okay, we'd love, as I said at the beginning of my talk, I introduced it as, remember I said human intelligence. Like we all really are motivated by understanding this. If you think about this, because we're the ones that are sort of owning the planet right now. We're a species that's special unto ourselves, so we're the ones who sort of want to understand ourselves. Taxpayers pay the bills partly for us to understand ourselves. That's not to deny all the other reasons of understanding biology, but this is really one of the prime motivating goals for a lot of the research is understanding humans. Okay, so I'm motivated by that question. I think many of you are. And here we go. Let's test you in doing this. And here's how we can test humans in general. Like, I'm going to fixate this dot. I'm going to show you an image. I'm not even going to tell you what kind of things are going to come up other than you probably already know there's objects, but I'm not going to pre-cue you and say you're going to see whatever. And then you're going to have a choice. And you can just sort of point left or right when you think you see what. Okay, what do you think? All right, look, did I tell you you were going to see birds and elephants? No, I didn't have to pre-attention cue you. I spatially attended cueing you there, but other than that, I didn't kind of tell you kind of what kind of categories you would see. In fact, you can do this quite readily with many different objects. Okay, left, yeah, right. So this is really just an operationalization of the thing I flash by. And that was showing there, I showed you a few synthetic images, and I showed you a more natural image like you might see in a computer vision. It's also a bird in that case. All right, but you see how naturally you do this, which is also a sign of like, okay, this is something that biology's figured out how we do well. We didn't do it well, we wouldn't be around to be talking about it. Again, it's like that Toiber problem of somehow we do this quickly. And again, I'm just demonstrating for you can do that. I'm going to this is a way for us to quantify how well you do it. You're not perfect, but also see both your successes and your failures, which are guideposts for us as we build models to say they should emulate those successes and failures. Yes. Right. So your point is, so notice if I had shown you this first, like your point is well taken, and let me just show you this. So here it is. So here I'm not giving you any side views, right? So you have an ability to do something here is all I'm really saying. Now, your point is like, well, actually, maybe your ability to do that is slightly to recognize this person here, which you're about to make this eye movement. You're fixating here, but you're going to do a little better in a situation where I show you this, let you make the eye movement, then you see the person, versus I just flash the person, right? That's the comparison that you're making. And people have looked at that. Gabrielle will speak about that, I hope, or perhaps. And the answer is yes, you get slight boosts from that. They turn out to be pretty small. You can find them. And they depend on the situation and all that. So I'm not denying that they exist. But the elephant in the room is like, wow, your system is so good at doing that, and that in just those flash glimpses. And that's why I'm sort of showing you that way that you can kind of isolate that as a sort of a powerful component right away. So I'm not trying to deny how this has to all be stitched together. And again, that's why I'm pointing to Gabriel. He's going to start to stitch that together for you. So keep that in mind and ask him that. I'm sort of punting. Gabriel, if you're here, I'm sorry I punted that to you a little bit. But I'm just trying to get you inspired by notice that you can do this quite well. And I think you can see that if you can do that quite well, the other problem, you don't have to do as much of some of the magic that you're talking about. So this was the problem that we operationalized and that we've been mostly focused on. So again, and I referred to it as core recognition. I'd like to quantify how well this system does it. which I showed you how we can test U. And we can test just not natural, but I also mentioned synthetic things like you might use or synthetic, but these are synthetic images where rendered objects are placed on random backgrounds. These are naturalistic photographs, which tend to have, we like these things because they eliminate covariation. Lots of live natural photographs, green in the back or blue in the sky, background might mean a plane in general. But we wanted to isolate objects from that. So this is one way to do it, is to synthesize things. That's why we did this in the past. I wouldn't dwell on this right now. I'm just trying to tell you, everything I'm going to tell you applies to both of these domains. Okay, the hard question about this, and this has been long known, and Tommy was one of the leaders in pointing this out. I think a lot of people have known this, but it's an old problem, which is identity-preserving image variation. So being able to recognize that's a car in all these situations, that's called the invariance problem. Being able to say that's a car independent of its viewing condition, including its background, which turns out to be some of the most challenging part. And the background interacts with these other variables here as well. And not just say it's a car, but it's a car and not a dog, or it's a car and not a horse. And that's the sort of core challenge. If you only had to identify side views of cars and side views of dogs with a perfect background, there is no challenge at all. So the challenge is being able to learn from a few examples like this and generalize across new examples with random views and so forth. Okay, so that's just getting into the computational challenge a little bit more than me just demoing you. Hey, look, you can do this. You were doing that. You didn't realize I show you those faces and they were tipped, and I still knew it was a face, right? And you'd never seen that face before at that angle. In fact, it's interesting to think you never, except in laboratory conditions, see the same image on your retina twice in your life. So we're always generalizing to some degree around what we've seen already. Okay. And then here's a measurement. Here's how well here's just a measurement of how well humans were generalizing. This is 2008. This is performance. So this, you can think it's better, higher is better. These are in psychophysical units of D prime. But accuracy, that's like 95% accuracy. This is as you increase the amount of uncertainty of the view condition, so more variation in the object. And you see that humans are pretty tolerant of this ability. They just kind of are almost invariant. I like to say tolerant rather than invariant because we still have a little bit of fall off. Machines at the time, they're really good in this regime here, which is this sort of side view of the car, always regime. They can sort of memorize those conditions, but generalizing to these kind of uncertain poses was harder for them, at least in 2009. So the core recognition regime was really, I think, 200 milliseconds here. This was how we were focused on this problem. How do you do that? See that gap between machines and humans? And I think this is also a general lesson. Finding gaps between current machine systems and human systems, where humans are especially better, is always a good place to sort of start a research program. And this was the one we were focused on at the time. This is just one example of where machines were below us at the time. You can think of the machines as the current hypotheses of the state of the art at the moment. You guys following me? So that's okay. Question in the back. I think I tried to unpack that logic there to say, look, when I show you this scene and I later ask you to, for instance, report, was there a person or a car or a dog or a sign? You're able to do that. Okay. And then I showed you the way your eyes move around means that this is what's hitting your eyes. And then I showed you, if I just replayed those sequences for you, you can do it almost as well. I didn't show you that. I was sort of related to this question. You can do it almost as well. So now it's like I can reduce the problem to cut out images of 10 degrees and sort of say, make that my problem, which is not still a full problem, but it's now operational and it's actually easier to study in the lab. So that was the trend. But I didn't go all the way to non-natural images because, at some point you go so far that you throw away the interesting parts of the problem. So I tried to unpack that logic for you, but I'm sorry if I skipped over something. That's one of the observations, is that our natural viewing time is just a couple hundred milliseconds. The other is that we have high acuity to center. It's a continuum. It's not like there's a 10-degree wall. So that's in some sense an arbitrary cutoff. And even the 200 milliseconds is a bit arbitrary because the distributions of eye movement time can range from as few as 100 milliseconds to you can dwell for seconds if you really try to fix it. If you look at natural conditions, for monkeys it's 200. For humans, it's actually a little longer, like 300 or so. But yes, those are sort of, I call that the natural operating regime of the system. And that, again, those are arbitrary engineers' choices of how to get started. Okay? So, all right. Okay, so, and I mentioned as computer vision systems at the time, you could say they were not as good, and they're still not as good at us in some ways we think, although they progressed a lot, in part because of the scientific understanding that's being built to help drive them. They're still not as good. So current humans are better than this, but we study in my group the rhesus monkey system, and so I need to show you a little bit about that. One of the reasons we're motivated to study the system, I'll show you here in a minute, is they can readily do the exact same thing you were doing. He's triggering this test image, and then choosing among two choices, just like you were. He's just using a touch screen instead of saying what he sees. And every time that means he got it wrong. But mostly they get this right. You might ask, how long does it take? It takes a few months to shape the animal and just be conditioned to this, kind of working in this little, you get used to the touchscreen. And then they learn a new object every about a day or two. So this animal knows something like 30 some objects. You can introduce new objects. And then they generalize quite well to new views of those objects once they learn them. And that's something we quantified in these kind of papers if you're interested. But what's quite cool is like one of these, and I've labeled them here, is the monkey's behavior, and the other is the human's behavior. These are sort of patterns of confusions over objects. That is how difficult it is to discriminate a hanger from a fork. I don't want you to dwell. These are like the D prime values. I don't want you to get into, in the interest of time, get into the details of how this plot's generated, other than you to look at them and go, I have a really hard time telling the difference between these. And that maybe shouldn't be surprising at one level because we were evolved from, this is a very close cousin to us visually. And they make the same they have the same patterns of successes and failures as we do. This is at the object level averaged over many images. We've also looked at this at the image level. You look at image by image patterns of what monkeys find challenging, what they find easy, what we find challenging and easy. Say, well, that was a car, but I'm not sure. And we can't ask the monkey if they're sure, but we can look at their behavioral probability of getting the answer right. And that's how we quantify the similarity and sort of accuracy patterns, even at the image grade. If you're interested in that, I can point you to a number of papers. These are some of the earlier ones. Okay, question, yes. I think you're asking a very interesting question. I think you're asking where the species, one question you're asking is where the species differences appear. Like there's probably some species, like if I ask the monkey, can you drive this thing? Like, they have no kind of, context of driving cars around. So there's certain questions they're not going to be able to ask. This is a pure visual discrimination. And so you're asking kind of how far can we push that? And that we don't know yet, but of course there's going to be some boundaries to that. But interesting, in this core regime, they seem to be very much like us. The second thing you're asking, which is also interesting, which is what about other questions like emotional content? Like, do you feel good or bad about this? That's actually something we're very interested in right now that I'll point to at the end of it. We don't have that much data on it, but we're interested in that because it has sort of clinical relevance to some things that I'll show you. But there's not that much data at sort of high systematic resolution. Getting a lot of behavioral data like this and comparing with humans is not a kind of standard thing that was done, and it's a bit of a grunt work to do it. But we took the effort to do it here because we thought it was important to quantify. But both your points are well taken. But I'm not going outside of those regimes right now. So don't read the talk saying Jim thinks monkeys equal humans in all respects. It's just with respect to these kind of discrimination tasks. Did I answer it? But please raise the emotion content thing again if we have time at discussion. If I don't, are you asking about? So we are not showing monkeys words in these examples, like written, but we have done that in other experiments, like letters and things. But I think that's not what you're asking. I think you're asking like the order, like when I give you an image, then I give you two choices, and like the two choices matter. Was that right? That is a good question, and we've done a little bit of that. There's a thought experiment of like, how well would you do if you were born and you only had reinforcement of juice and you have no language on those tasks? So we start to ask the question, but then the interesting form of that starts to be, how do we compare it with human abilities, right? And so then we have to sort of imagine your trajectory of learning and so forth. And we've dabbled in that, but the answer is we don't fully know, except that they can discriminate letters and even subsets of groups of letters that form words from other things. That's also been demonstrated in other primates like baboons. It's not something I'm going to talk about today. They can do it to some degree, but comparing them with humans in these this is called orthographic processing and the introduction to language, how well they can do it and exactly match humans is not. I think the boundaries of that are unclear. It's one of these related questions, like where do the limits start? And so the answer is we know a little bit, but not a quantitative comparison like I've done in these cases here. But I could point you to a few papers if you're interested afterward. Yes, I see a question there and then maybe we should move on. Yeah. Well, that's what we're going to show you neurally next. So to jump ahead, the neural data for the areas that we think are involved produce the solutions to these objects at the level that humans and monkeys do them in about 200 milliseconds, some cases a little longer. And we'll show you that. That's a lot of where the front line of research is. And I don't mean to imply it's all feed forward, but it's pretty fast, okay, for doing these kind of things. Which, if you think about it, the system is evolved to do this thing. You want the system to be producing decent representations. Otherwise, it should slow down its eye movements. So the machinery and the eye movements are sort of paired in a way to work in that regime. You can actually break that, by the way. You can move the images to you, and you can show that you actually can do things like read faster if someone else is moving the words for you than you having to move your eyes. So in some sense, it's not the visual system that's limiting you. It's the movement of the eyes that's limiting a lot of your processing. But the system is quite fast, and I'll show you some of the data to support that. You should already think it's quite fast because I just demoed for you. Look how fast you can do this behaviorally, and that's been known from work of Molly Potter and others all the way back in the 70s. You can process that to content, not all latent content. When I say latent content, I mean the things that are underneath generating the images in the world. Like there's a car actually out there in the world that's causing those photons to strike your eyes. Or there's a sign back there. Notice you can't everything, but you can get a lot. And I'm focusing on that part that you can do, which is sort of the foreground object and its latent content, especially its category or identity. Yeah, again, we are just starting to do that work, so I don't even want to say much about that. Not because I'm hiding something, just because I don't know that much yet. You're asking another question, which relates to just, let me simplify this, which is like, if I give you longer viewing times for just this task, forget emotional judgments, just for this task, how much better do you do? Here I'm giving you, and I don't know if I inserted that plot in here, but it's like you get a little bit of gain, but most of the action happens in the first couple hundred milliseconds, right? Which, again, if you think about it, it's nice the system's tuned itself that way. But I can show you a little bit better when you go longer. So again, that's sort of in the spirit of the kind of question over here about, hey, is this all you can do? And the answer is not quite. But again, a lot of action happens already in that. So that's why I'm focused on that. Yeah, again, a lot of questions now, which is probably good, but I want to make sure we keep moving. So, and I think I skipped, somebody else was next over here. Yes, go ahead. So random shapes. So sometimes you mean things that have, you don't mean random pixel sets. You mean things like that have coherence to them in the physical world, but are configurations of parts maybe that you've probably never seen before. So we actually did. Michael Lee is a PhD student in my lab, just defended his thesis, essentially studying human abilities to do that. He actually get monkey data on doing that too. And you can do that pretty well, and so can monkeys, is the short answer. But it takes you a little bit of learning time because you got to learn what objects is what. You got to start to name them. But yes, you could do that pretty well. And he showed that the representations that coming from the models that are best models of IT they appear to be sufficient to support your ability to do that. So again, the way we conceptualize the world is you produce a feature representation from the images that might be feed forward, might be recurrent, some of both. That feature representation is going to be followed by a simple classification, and that supports your ability to do these kind of tasks. That's what I'm going to all tell you next. That model with the right kind of feature representation supports even that kind of task. And that's kind of what his thesis was about. At the same abilities of humans, the same learning rates of humans, you don't need anything more than a good feature representation to do that. But that's getting into his thesis, and I don't have all the slides for that. And maybe you can hold that part about novel objects, and we can return to discussion at the end. But I think this applies to that question. I think these models I'm going to show you apply very well to this. It's not as if, oh, these are only well-learned objects. In fact, I think you pointed this out, as monkeys, they didn't know what a car was until we started teaching them in the lab, presumably. They might have, they're usually just in the lab rooms. They don't see cars. They're not out on the street. Okay, so these are all sometimes novel to them when they learn them. Yeah, that's another good question. Gabrielle's going to talk about that for sure, which is like how you have a salience map thought to be computed mostly by the dorsal stream. That's the way we think about that. That sort of drives your eye over there. And I think what we're telling you is like when your eye gets there, information is being provided and estimates are being made at the center of gaze, which is what I'm talking about. So that's in some sense why my talk is preceding his, but hopefully you guys can ask him that question as well and how he thinks about stitching that together. That's exactly the kind of thing that he's going to talk about. Because your world is not just stare at central 10 degrees and have stuff coming at you, right? But you have to still decide where to move those eyes, so to speak. Which is the question you're referring to, which is a good one, but not one I'm. Okay, one more and then let's move. Okay, so that's, you guys are all, I'm putting everything on Gabrielle. I'm sorry, Gabrielle, if you're listening. Okay, so as I mentioned, neuroscience we're interested in human, but the monkey, we study it partly because they're really matched to us in their abilities, as I briefly outlined for you. But decades of neuroscience, Tommy mentioned Hubel and Wiesel, really back from the 60s, have started to lay out for us the areas that are involved. And we know a lot about the functional properties of the neurons and the anatomy of these spaces here. So let me show you sort of a quick tour of that. So these areas, these are cortical areas. We refer to them collectively as the ventral visual stream because they're on the ventral side of the brain. They're both feed-forward connected and feedback connected, as illustrated here. And this is a little bit like when I sort of joked at the beginning. I said, well, I could just tell you that the ventral stream solves object recognition, write that in a textbook, and we could all go home, right? And that's like, hey, my iPhone does this app. What's the problem, right? So this is what you'll find in the textbooks. But the problem is, okay, how does it actually work at a more detailed level, right? So, but these we knew sort of lay of the land, what area is important. We know this in part because lesions, as I write here, lesions here, especially in these upper regions, produce deficits in task-like recognition in both humans and monkeys. And this highest level is called IT, or infratemporal cortex. I'll talk a lot about that because it has very powerful representations. It's sort of the last mostly visual representation before you start getting into very multimodal representation. So it's sort of the top of the visual processing chain of the ventral stream. Here's the rhesus monkey. Let me just show you how this is now to just tell you kind of how we know this. I'm just going to do this as a whirlwind in the interest of time. That's a monkey brain. You can take the monkey brain and slice it up, and here's like a cut through the monkey brain. You can start to see the layers in the brain where the neurons are occupying. Here's a zoomed in 6-layer cortex. You don't know cortex has 6 layers. Each of these dots is a neuron. And you can see the neurons have different sizes and densities across layers. This is partly how we organize cortex by the differences in this anatomically. Neuroscientists refer to these as layers. Those of you who play with ANNs, you'll also hear the word layers. That's a confusion in the field. Layers in ANNs, as we map them to the brain, are like areas in the cortex. So there's a language terminology. So this might be area V1. Here it's 6 layers. This is just to kind of orient you. Now, these are the cell bodies. The cells, as you probably know, they're not just dots, point dots. They have big processes. Dendrites, like these are pyramidal cells. See, they have big, long processes coming up here. And they also then have axons extending out of them. And there's lots of interesting processes that happen within the neurons. And that's an active area of study, too. In the models I'm going to tell you about, we treat them essentially like point processes, which means it's sort of just an approximation of their integration, which is more complicated than that. And the data that I'm going to tell you, a lot of the physiology data comes from just parking electrodes nearby neurons, just randomly sampling and listening to the extracellular voltage changes that are generated in response to the action potentials within inside the cells. So we're going to this is an action potential. It's about 0.5 millisecond long in duration for those of you who haven't seen action potentials. Has a special privileged role in online vision. Why does it have can anybody say, why does it have a more privileged role than, say, calcium levels in the cell? Anybody want to? It's not a trick question. Like, this is something we can record. You might be like, those scientists, they just like to record. Whatever they can record, they're going to tell me about. They can measure fMRI blood flow. They're going to tell me about blood flow. They can measure genes. They're going to tell me about genes. With regard to the vision questions I set up, can anybody say why I think action potentials are especially an important level of phenomenology? Not just that we can get it, but why they're important. I saw one. Sorry? They're fast and they're transmitted fast, right? So they can get, so it's about 1 meter per second, but about 10 milliseconds per cortical area. And the only way we think you're transmitting information at the speed of vision for 200 millisecond operation is through these signals, right? So that means we're sort of tapping the main signals that are being propagated through the system for online vision. Now, if you're interested in, that's for questions of online vision inference models, how they work in real time. They're special. If you're interested in learning, you might want to sort of remember this is not just a special level. All those other things come place over hours or minutes. You should not think of them as quite as special, perhaps. So that's why I'm pointing that out. I'm focused on this. I'm going to tell you about models of online inference processing, and that gives these a privileged role here in building models of that type. Okay. I'm not going to talk about models of learning. I'm going to talk models of visual inference or visual processing, if you prefer. Okay, so how do we know one area? You've got these areas I showed you, V1, V2, V4, IT. How do we know one area is sort of above the other? Well, the short answer is a bunch of decades of studies have shown like neurons in one level, like layer 2, 3, tend to project to layer 4. That's called a feed-forward pattern. Other levels, like 2, 3, and 6, project backwards to these upper and lower levels. It's called a feedback projection. And so by ordering a bunch of cortical areas over many, many years, this is a kind of well-known layout of a hierarchy of the ventral stream, sort of based on anatomy of the connections between the different areas. We can think of one area as being higher than another by ordering what many studies of pairs of areas like this. Also, the dominant input to the cortex arrives from the LGN into area V1, so that sort of sets the start point, and then the anatomy frames the rest of this. The latencies of these such are about 10 milliseconds later for areas you go up the hierarchy as well. So that's functional data. All of this sort of kind of motivates a general hierarchy of visual processing. Here it is. This is another version from one of our review papers. I like this version because it kind of gives you a sense of the scale of the areas. I drew this where it's sort of sized of the number of neurons approximately in each area. So retina LGN, about a million cells in the retina projecting out, retinal ganglion cells. V1 looks at hundreds of millions of cells, so a big expansion in terms of cell count. V2 similar. I've colored in red. Oh, this relates to the peripheral vision question. I've colored here a fraction of the tissue that's devoted to the central 10 degrees. So in case you think, oh, central 10 degrees and we see 140, well, look what your visual system is doing. It's like that stuff is not well represented in a primate visual system. That's because your retina has got most of its power packed in those central 10 degrees. So even though it sounds like a narrow part of vision, that's actually most what's going on here, what you see in these colors. Okay, V1, V2, V4, IT. I'm showing you the feed-forward flow, the approximate number of neurons. You don't need to memorize all this. This is just to give you context. You might have heard about MT or other motion areas. Like, look how tiny they are compared to IT. So it's sort of a lot of visual systems devoted to this process, and this slide makes that clear. Okay, this is just tour anatomy and like why we even talk about the ventral stream. I'm going to tell you about infratemporal cortex. So I already mentioned meso and macro architecture. Tell us about the feed-forward feedback. You don't just produce IT neurons and then like, okay, that's the end. Somewhere those brain areas connect to other brain areas. So to orient you, IT projects especially to frontal areas involved in decision and action and around the bend to the medial temporal lobe and areas involved in storage of long-term memory. So, and that seems like well positioned for things that would be like object-like representations, either act on them immediately or store them for later use. And so this is just how neuroscientists have sort of gotten the lay of the land here, just to give you a sense of mostly visual processing and then some stuff that sort of feels like it's beyond just vision after IT. With me? Okay. Okay. I'm an engineer by training. I like to not think of these biology tangled up sort of cortex, and I showed you all those cells, but just to say, well, look, think of this as millions of neurons in each area, and I gave you the approximate numbers. And here I'm just going to draw them as sort of conceptual drawing of millions of neurons in each different area. There's some transformation, and when this slide is a bit old, I'd always like to think of whatever transformation might be in common between cortical areas. Somehow it's some magic transformation that's built in. You can ignore that T for now. It's sort of an old slide, but basically the key idea I want you to get is there's feed-forward flow, there's recurrent flow, there's recurrent inside an area, and there's recurrent from other areas. There's also skip connections that I'm not showing you here. So the system is more complicated than this first order drawing, but these are the dominant anatomical connections on first pass. Okay. But millions of neurons in each area. When I show you an image like this one, this is just to set up this conceptual idea. It produces on your retina essentially a photographic copy of it. So now you've turned light patterns, photons, flux on your retina into spiking rates out of retinal ganglion cells in sort of a pixel-wise manner. It's not completely pixel-wise. My apologies to my retinal colleagues. A lot of interesting processing happens in the retina, but it's sort of still very local in terms of its sensitivity to light changes with regard to sort of saying like the size of an object in this image. So think of this as a so-called isomorphic representation, like you might get out of a good camera on your iPhone, and it's a pixel-wise representation, but it's still neat. We haven't yet extracted so-called latent content from it. And we think that that's what happens down the rest of the system is that you're getting sort of successively progressed patterns, new patterns of that population activity that takes about 100 milliseconds to reach up into IT cortex here. I'm showing you here a very rigid feed-forward flow. It's not that simple, but these are the average latencies suggest that that's sort of, again, a first-order approximation. And a key thing I want you to keep in mind here is don't just think about one neuron anywhere, but think about populations of neurons. They're the ones carrying the information. So population carries, you can't do anything with one pixel. Similarly, even up here, you don't do anything with one neuron. Think about a population of IT neurons, perhaps the whole of IT, supporting how you're re-representing the world for those other downstream brain areas. And the challenge for us has been to really figure out especially what's going on along this chain here. That's what I mean by engineering spec diagram. So these are conceptual patterns of firing of neurons is what I'm trying to illustrate. If I didn't say that, like the colors lighting up means that neuron's firing or not. I'm showing you in a binary way. Of course, it's more complicated than that. There's gray. It's not just on or off for the neurons. But this is to set conceptual ideas for you. Yes, question. Right. So I think you're asking like what we want is continuous time input models. So here, of course, if I think about that image, if you plot it across time, it's like it's nothing. Then there's a step function for some number of frames. Then it's off. Right. So there's time always. Even though we call it a static, there's always time. Even when I show it 100 milliseconds, it's just that's the time profile of the input. It's a step function for 100 milliseconds or 200 milliseconds. And what you're saying is we would really like models that would take temporal input and produce temporal dynamics. A lot of the models I'm going to show, some models do that now, and I can tell you about some of them. A lot of the models I'm going to show you today are really just taking one input, like that input, process, And that is a very useful start point, we think, because it's focusing on the spatial content of the image without the temporal, worrying about all the temporal effects. But the more recent interesting stuff has to do with temporal effects. And Ko's work speaks to that a lot. And if you guys interact with him, he can speak to you about some of the latest. So yes, that is an ideal. But I'm going to show you today, just getting the spatial models seem to be a sort of good starting point. And again, our intuition, or my intuition when we started this, is like this problem is really one of spatial processing mostly as the sort of core interesting problem. Notice how I keep all these questions, I keep narrowing the clouds. Like, oh, we were going to be like all of intelligence. No, we're going to do this. We're just going to do these spatial models. And that's, I hope we don't narrow it so far they're like, wait, now it's not interesting anymore. So it's good that you keep pushing me on the edges of this, but you need to do something like that to get started. And so these were choices that I think were reasonably wise to focus on space and not time. But your point is well taken that we should be striving for temporal models. And that is kind of active work right now. Did I answer your question, or did I just spew off random? Is that just random stuff? Was that helpful? Okay, yeah. Yeah, so you're asking, I think these are very interesting questions about how many units here, and you're referring to dimensionality. So this gives you the so-called ambient dimensionality, which is like how many physical units you have. But the interesting dimensionality questions live beneath that, right? This sets an upper bound on the dimensionality. But if a lot of these neurons are doing identical things, then the functional dimensionality can be much lower. If you follow that, imagine every neuron of V1 was identical copy, it'd be a dimension of 1 functionally. So we don't actually have good estimates of the dimensionality of these areas. We've worked a lot on the estimates of dimensionality up here. And even those are hard to get, but we think this order is sort of strange. It's on the order of like 1,000, maybe hundreds, even though there's about 10 million output neurons. So there's a funny like redundancy factor in there. And that might lead you to say, well, maybe those neurons are doing something else. Maybe you're estimating dimensionality wrong. But we don't actually have very good estimates of the functional dimensionality on these areas. But I share your intuition that it's probably going from higher lower. So there's a step up here and then a shift down. And a lot of the neural network models sort of emulate that kind of idea too, which is sort of like you blow up the space, think about pushing all the images into a bigger dimensional space, and then you reassemble it to something that's lower dimensional and better formatted. Because if you stay in high dimensional space, it's very hard to learn. You're not going to be efficient in that space. Even if you're separable, you're not efficient for learning. So you want to reduce dimensionality to a space that's likely to be problems that you are going to encounter in your world. And that also means you're throwing out stuff that you're not going to be able to learn. And I say humans are powerful, but somebody asked me about novel objects. If I show you novel random pixel patterns, you're not going to learn that very well. So I could call that a novel object and say humans don't learn, but that's why whoever asked that question, I was pushing on that aspect of what you mean by novel object. Sorry, that was a long diversion from your question. Did I sort of answer? Yeah. Okay. We're getting lots of them, but this is good. Okay, let's go here. So I think of the connections as separate from the number of units. I thought this was orthogonal to connection. You could still have a high dimensionality and skip connections, a low dimensionality and no skip connection. Those are kind of two different issues to my head. The connections just shape the functional properties of the neurons is how I was responding to this question here, but I'm therefore giving the neurons precedence as the fundamental units themselves. So I'm not sure I'm answering your question, but maybe if I'm not, we can return to it at the end. Yeah, let's save this question. I haven't actually showed, for those of you who haven't seen ANNs, maybe you all know ANNs are best models of this, certain ANNs are best models of this. And there's a question of what's the mapping? Is it a one-to-one mapping between actual units in an ANN and actual units in the brain? Right, and that's sort of part of the spirit of your question. And the short version is we think there's like 100 to 1,000 to 1. When you look at most neural networks, they're like 100 to 1,000 times smaller in terms of number of units than the number of neurons in the brain that I showed you here. So I like to think of that as just biological redundancy. And they're all, think of them as all copies of the same thing. Now, they're not going to be exact copies, they're just in the same linear span is the more precise engineering way to think about it. But there is a factor of at least 100 relative to today's models when those things are mapped. So that is another interesting forward question about how these models relate in exactly the way that you're pressing me on there. Let me move forward, unless there was another one that was really burning. Oh, yes, sorry. Oh, that's a good question. So when I draw these, I'm tending to draw what I think of as like the layer 2, 3. When I draw this right here, and I'm drawing it as one layer, each of these areas is now down to one layer, even though you're right, I said there were 6. The way I think about this is like this is the output of V1, which is to V2, which is mostly out of layer 2, 3 from that early anatomy I showed you. So think of this as the number of layer 2, 3 cells. And I'm not actually, so see the differences here between these numbers. See V2, it says 150 million. and then a V2 representation says 29 million. That's an estimate of the number of pyramidal cells in layer 2, 3. So you can think of the rest, I like to think of the rest of that as being part of the processing to get to the output of V2. And, it'd be great if we knew all our electrodes were recording just the output neurons to constrain these models, and the rest of that sort of like think of it as a cortical black box, right? It's like, hey, something happens in V2, and then some outputs come that are being processed down out of a V2. That's back to this timing thing I mentioned about spikes, right? Okay, I got to get that information up to V4, and I only have a few milliseconds to get that done. So I want to record, that's the signal I want to record as the engineer, just that output level as a start point. In fact, the electrodes end up recording multiple levels, but they tend to record the big pyramidal cells, especially in layer 2, 3, and we try to target that, but we can't guarantee that yet. That was another diversion from your question in experimental data, but yes, I'm showing you layer 2, 3. Think of it that way. That's what we're trying to model. Let me just kind of, in the interest, I have 10 minutes before we're supposed to get to break, so I'm going to try to sort of, I know this is all, but this is all helpful, I hope, to you guys. Okay. So how do we know this? I've already sort of alluded to, how do we know about these firing properties? Well, remember, this is a monkey. The monkey can just stare. Now, I showed you monkeys doing tasks. We get a lot of our data from monkeys just fixating, like bringing the images to the monkey. The monkey's just fixating, like passively. Turns out a lot of the processing that we measure seems almost identical. Co can also speak to this, almost identical in the passive versus the active recognition situation. Kind of like the machine can't help but run when that photons strike the eye, at least the machine that we're measuring here. Now, again, all things are not quite exactly the way, and Co can speak to the differences there because he's recording both active and passive conditions. But think of this as just passively viewing data, as a lot of data I'm going to show you. And we go ahead and stick an electrode in there, actually multiple electrodes I'll show you in a minute, and we sample neurons randomly from these different cortical areas. In this case, here's a sample out of IT. Those of you who haven't seen neurons spiking yet, these are tick marks, indicates a time of an action potential. Remember, that's an action potential. The stimulus is on for, in this case, about 100 milliseconds, and then you see there's a latency of about 100 milliseconds before you get a bunch of spikes. I'm going to show you that that's actually reasonably reliable. Notice it's not lockstep. There's spiking variability here. But that's a sort of typical kind of transient response you get out of an IT neuron, a response to just one example image shown to the monkey here. This is repeated randomly many times, and I've just stitched it back together. Each row is a presentation of the image. You guys familiar with time histograms? That's what this is. This is just a raster plot. Okay, those are spike times out of one neuron in IT. Just to familiarize with the data, here's another image. See, this neuron seems to respond a lot to this image and to this image. Not so much, a little less. See, it's like a little lower here. You're sort of integrating with your eyes. So taking a mean rate, high mean rate, medium mean rate, kind of lowish mean rate. Those are actually the measurements that we use for comparing models as the mean rates. We're not tracking the individual spikes. And that's another interesting topic going forward. But here I just want to give you a sense of the differences in mean rate. Here's another site. Look, it doesn't like this one as much, but it likes that one, and maybe that one a little bit, and this one a lot. So different IT neurons. Here's one. Oh, I like these two images. We think of these as just different neurons like different images. If I had shown you this, I could have maybe written a paper 20 years ago and said, oh, I found a potato neuron and a something neuron because I had these plots. But I like to remind, these are just kind of, we think of them as just responses from images that are more complicated than potatoes or so forth. And that was actually part of the challenge as to how do we model that. So, okay, I just mentioned this. This is the mean rate. We're going to actually just tell you about just count spikes in a 100 millisecond window here. We changed that later to be more time narrow. Again, a good question for code to talk through about. But here I'm just going to tell you about 100 millisecond window. And we latency adjust for, remember, the system is not zero lag. So it takes, I mentioned this briefly, about 100 milliseconds for the information to go into IT. So your eyes are already getting ready to move to the next stage place while your ventral stream is still busy processing the last image. Interesting to think about, right? Because you're moving your eyes here and you're still gathering the information, but the information's already been pushed into the system, into the pipeline, so to speak. Okay, so we can go ahead and now measure for each of these images. Like there's a different population pattern that I'm illustrating for each image, and it can be generated even at these rates that I'm showing you here that are in line with your kind of core recognition abilities. So now we can go ahead and record those data. In this case, not with single electrodes, but with multi-electrode arrays. And the reason we do this is twofold. It's like we can get more units per unit time. So in this case, we get like several hundred units simultaneously. These are implanted during a sterile surgery. That's the monkey's temporal lobe IT cortex. That's the dura mater. That's the skull. They're implanted during surgery. This all closed up. The animal has a connector on his head. We can plug the animal in every day. This is the other reason we really like this is because these electrodes are quite stable, often for many weeks or often months. We can record the same, at least multi-unit sites for a long period of time, which means we can show many images, thousands of images for many reps. And that gives us much more power on sort of fitting models to data. Okay, so here's an example. Conceptually, what I want you to think about is the idea that now it's not like the previous slide I showed you 1 neuron in response to 4 images. Now I'm showing you many neurons in response to 1 image. So change the axes on you. So this in computer vision we call a feature vector, sort of the response rate from each neuron. I'm showing you a few of them. There's neuron 1. These are just arbitrary samples out of IT. I just ordered them arbitrarily here. And all I want you to see is sometimes the neurons like the image, other neurons don't like the image, or that neuron likes the image, just like the examples I was showing you here, just ordered by in response to a particular image. This is to just remind you that, remember, it's a population that we're interested in. It's not like one neuron is doing all the work. It's a set of neurons. So this is an estimate of the population. Notice the population changes its output like these. This is conceptual. These are real data. The neuron's changing its output. The neuron set in IT is changing its output in response to changes in the image. So you get these different feature vectors out in response to each image. This is just empirical data to give you a feel for how we get some data to constrain models. Guys with me? Yes. Yeah, so this is related to the layer question. We build our electrodes. We choose the electrode lengths so that and we hope that they're in the upper layers, but based on their lengths and the thickness of cortex. That's what I said earlier. I can't guarantee what layers we're recording from. So I mentioned that earlier. So think of this as a random sample, mostly biased to the upper layers. Also, these are often multi-unit sites, which means there are multiple neurons living nearby. Luckily for us, nearby neurons have very similar functional properties. So, but we're treating them, I call them neural sites if we were honest about it. Now, often that's what you'll see me say, neural site. I think I wrote that. Sample number site. So they're higher resolution than like ECOG, much higher resolution than that, but they're not often quite single neurons alone. We can isolate single neurons, and everything I tell you when we look for isolated single neurons, we don't see differences in those things, but it's much harder to get a lot of single units. So multi-units sort of the way we tend to go. But yes, we don't know the layer of them. We think they're biased upward. Okay, in the interest of time, okay, here's one of the main messages I want you to get from this first hour here beyond now we're on the same page about background and what the heck is DiCarlo talking about, I hope, is that there's this population that you can record out of IT. Conceptual idea, but you can empirically sample it. This is an example of an empirical sample. Many thousands of images, many hundreds to thousands of sites. That's what's on this plot here. We repeat many presentations, like 20 times or more per image, so we can get very high SNR into the mean firing rate. So that those are the core data that we obtain when we do this. And what we can show is that when you take linear decoders and you say, give me a few examples of cats and a few anti-examples of cats, and you build a linear decoder, and then you ask, so you're reading IT as a decoder, and you're asking how well does it predict what the performance accuracy on cat versus not cat will be on held out images of cats. It does a certain number. That number tends to be well above chance, and almost, when we choose the parameters right, almost exactly aligned with the animal's behavior on saying it's a cat or not cat, back to that behavioral data. These are the behavioral data I showed you earlier, just to sort of give you that conceptual idea. But a set of linear decoders trained with on the order of tens to hundreds of examples of images tested on held outs produces the patterns of errors and successes and failures that we see in the monkey that, remember, was matched to the human. So again, a set of linear decoders on IT reading mean rates seems to be sufficient to explain the animal's behavior. If you do this on the retina or pixels or V1 simulations, you do not get this result. So this is a big, this motivates a couple things. Oh, IT is important. We already thought that, but now here's a decoding model of how you go from IT to behavior. It gives you parameters on that model. It's a possible falsifiable model. It's not just saying IT supports behavior. It also says mean rates tend to be sufficient over the time windows I showed you, at least at this resolution. It doesn't require spike by spike times to predict the animal's behavior. So there's that result sort of kind of supports a lot of implicit assumptions that I've been setting up for you. Nothing's guaranteed in science, but at least say, hey, look, if we had downstream neurons reading from my mean outputs of IT neurons, they could be what's the animal's using in its head to make its choice. That's the sort of bottom line here. Linear reads out of IT seem to be sufficient to do that. Tons of details on how we analyze this so-called decoder models, and there's a whole lot of open space. It is not a done problem, but it sort of says, actually, you're kind of on the right track here, it feels like, because this seems to be sufficient as a model. At least that's how we think of it. This is work actually I did with Tommy way back in 2005. We started this, and Gabriel Kryman, who you can also hear from. We sort of showed that if you just build these linear decoders out of just reading 100 milliseconds out of IT, again, that's a feature vector, just like the green one I showed you. You build linear decoders on that. Remember, if you guys, hopefully you've had linear decoders, you're going to see that today. Linear decoder means you have a space where each axis is the response of one neuron, and there's some n neurons. Each point in this space that's the feature vector. That's the response of all neurons recorded to each image. So each dot is an image. Here's 3 example images. They're labeled by their ground truth class. In this case, it's just conceptual. Green means face, red means non-face. The idea is you build a line if this was 2 space. It's a plane in 3 space, or it's a hyperplane in higher dimensional space, which is what you're seeing here, which is just partitioning the space into positive and negative examples. You have to train the position of those lines. That's called training the decoder. And then you test on held out images, which are basically going to be dots in this thing. And there are going to be certain patterns of successes and failures. And what I'm telling you is the data so far suggests that those patterns of success and failures match the animal's performance patterns. So what that suggests is the animal in its head is building linear decoders, something like linear decoders, which actually downstream neurons can easily do, is thinking of weighted sums and thresholds. That's what a simple point model of a neuron does. That's how we think the animal is learning objects, or how you might learn objects, is build some fixed set of decoders on some example data, and then hold those as that's my set of cat neurons, my set of dog neurons. And that model, it seems to be sufficient if you're reading those out of IT, and generalizes well over things like position and scale and all the stuff I said at the beginning. That's a sort of big take-home message from a number of papers. That is sort of the context in which a lot of what I'm going to tell you next motivates. Like mean rates out of IT is an important target because it can support these kinds of behavioral analyses. The decoder models seem reasonably sufficient. Now, again, I'll point to Ko if he's still here. Those decoder models are not perfect, and we can see when they deviate slightly from the behavior. And now we're pushing on, well, maybe we got to get a temporal model to fix those, and that's actually an active line of work that Ko is working on. So don't consider these models done. They're just very good start points to ground us on like how it might be working. Okay, like all science, this is never finished. This is just our current models. Okay, I just said the parameters of these decoders. You might say, why do I care about these decoders? Well, they tell us how you might manipulate IT responses to modify object percent. So any good model, and you can think of the decoders as a hypothesis or a model of how IT supports the behavioral choices, should offer predictions. So the kind of predictions that these models offer would be, if you perturb these IT neurons in this way, here would be the change in the animal's percept if you did that, like injected current, silence neurons, that kind of thing, right? Those are the kind of predictions that decoder models make, because they're linking models between IT and the animal's behavior. You guys following me what I mean by model? And actually, we've done some of these, and others have done these experiments. When you start to try to do those experiments, so far, these seem to be as predicted. I say approximately, because these are pretty hard experiments to do, and their strength of alignment, we can't play the piano in IT cortex. It's a short version. We can't turn that around, turn that. We can't do that. So it's very hard to sort of strongly test the predictions of these models, but they seem to be roughly in line when we do things like silence groups of neurons or activate groups of neurons. So we don't have a reason to reject them from those causal experiments. And if you're thinking about BMI or like inducing percepts, they should offer, here's exactly how you might think about doing that if you had the tools to sort of play the piano on the neurons, so to speak, but that requires a lot of technical tools that we don't yet have. But I'm just trying to kind of give you a sense of what these engineering models in principle could do. The decoding model is one type of engineering model. The rest of the talk that I'm going to have after the break, we're going to talk about is like the rest of this here. And I'll just sort of set this up here. Is like even if you accept all that, and you might not, and you might push back on it, but if you accept all that, this is relevant for AI because you can think of a big summary of what I just told you this morning, is that primates are behaviorally better than humans or computer vision at the time because somehow their brain produces this good representation that computer vision systems were lacking. Because if they had it, you could just build linear decoders and they should look like monkeys. But this sort of implies they didn't have it, or else they would have done that curve I showed you where they were bad. They just didn't have this representation. And so that was one way to interpret that. And another science way to think about this is, hey, my goal isn't to explain every spike, but just to produce the mean firing rates at, say, 10 to 100 millisecond resolution. If I can get models that can do that well along the ventral stream, then they should be a good start point of the models we should be most focused on. This is the behavior of interest, in this case, core recognition behavior. Sort of ground that question. They turn the question from explaining behavior to explaining these mean firing rates out of patterns of IT. And you can sort of turn your attention to models of that type. Now they're just aiming to explain neural responses. Of course, you'd also want the model to then explain the behaviors, so-called end-to-end, but it allows us to sort of break the problem in two, if you will. So-called decoding from IT and encoding from pixels to IT itself. That computer vision we call the latter the representation, and the second one is the sort of the classification step. You guys following me, or am I just rambling on? Okay, so I'm kind of telling you kind of why this is all like history. So, okay, what is the integrated set of mechanisms across here that I've been showing you? That's always been the holy grail question for us, because we already knew that IT was pretty good. These neurons were pretty impressive, even before the work that I did with Tommy and Gabriel. We love that work, but history had already showed us ideas. So this wasn't like in a vacuum. We knew IT neurons had sort of powerful properties from prior work. Desimone, Gross, and others had shown those kind of things. Okay, the rest of the talk, I'm going to talk about this. I will just set it up, and then we'll take a break. I think I have the break slide in a minute. I'm going to talk about this question. How is this response being produced from the pixels? What I call an encoding model, online vision, like in 100 milliseconds, how are you producing this across here? Notice this is a how is it being done question, as opposed to all kinds of questions in science. Right? This how does this go to that? It's not a normative question. Why did it choose to go this way? It's just a how is it being done is sort of the way I think about this. Okay, it's not a question of learning. It's a question of there is an adult monkey or an adult human that somehow does this. I'm recording it in the lab. How is that working in that adult? It doesn't mean I'm trying to necessarily understand a full developmental trajectory. That would be great. It may provide a way to get there, but it's not the core question I'm asking here. I'm just trying to find a version of the system that does what the biological system does. That makes sense? So it's just separating learning from inference if you're using the sort of machine learning jargon. Again, I just said this, not evolution, not development, not learning. That's not saying those are not interesting questions that are not relevant. It's just helpful to keep these two different questions in mind so we're not talking past each other. And our goal, the lab's goal really for the last 10 years has been to try to find, build, acquire by hook or crook, whatever way we can, an accurate engineering model of what's going on in here. That's been our goal, right? It's just to get that kind of silicon version of that thing. Okay, and I'll motivate for you in the next part to talk why that's useful beyond understanding learning, because it's purposely not an attempt to understand learning. It's an attempt to find a visual system copy, if you will, in engineering terms. You guys, I see some nodding. Are you guys with me? I'm just trying to sort of set this up. Okay, how do we proceed to do this? And this is going to set up the break. We're going to need some kind of system model back to the beginning that has neurons, multiple neurons, that has multiple levels, maybe have some recurrence, maybe some skip connections that go from pixels to neurons to neurons to neurons to more neurons. That's a system model. Oh, that's called an artificial neural network. So that's how we get to artificial neural networks. That's why we use artificial neural networks, not because we're like, oh, no, AI, artificial neural network. No, we've been here for a while. This is what biologists, this is the way the question looks to a biologist. Before computer vision decided ANNs were cool, this was always the question. Okay, so I think just setting that context. And then I'm going to just motivate you to remind you that these IT neurons, we did not know how they worked. Like even as early as 2013, we still don't know exactly how they work. But we had no clue about how to build a model that would predict them well. If you show images, this is now the response of an IT neuron. Actually, you know what I'm going to do? I'm going to pause here because this is going to better motivate after the break. So, and then we'll go next. So that seemed like a good place to break.